This is a demonstration of the translation process. It happens in the cytoplasm of the cell. So we have 20 amino acids and only 4 nucleotide bases in DNA. So we use triplet code, which is 3 nucleotide quote unquote words. They are just letters put together. It's non-overlapping and is used to code for information from the gene to transfer to protein. So we have 64 triplets. 61 of them will code for amino acid. 3 of them will code for the stop signal. The triplets are called codons, and they code for individual amino acids. So one of the question is what sequence would code for MET, THR, ALA, TRP, and stop, right? I made a little chart right here. So methionine is the codon will be AUG and it codes for start. Threonine, we have ACU, ACC, ACA, ACG. For alanine, we have GCU, GCC, GUA, and GGG. For tryptophan, the only codon it has is the UGG. But for stop, we have 3 different types of codons. We have UAA, UGA, or UAG. The other question is, explain what the significance of a universal code is. The universal genetic code, it is significant because it supports the theory that all life evolved from a common ancestor. And up top, we have a demonstration. It shows the mRNA and the process of how the ribosome moves through the mRNA and how the polypeptide is made. So right here, this is the ribosome.
Big mushroom.
So the codons, they're the ones that code for each for individual amino acids. So we have amino acids that are binds to a transfer RNA that binds to a anticodon. The anticodon determines the different types of amino acids, and they would bind to the codons. So for UGG, U, it would bind to A, G will bind to C. So we have the anticodons would be ACC. So this is how ribosome moves. So I'll just put this under here.
Big mushroom. Big mushroom.
So we have small ribosomal subunit and the big ribosomal subunit. So in here, the charged RNA molecule will bind to the A site. So we have this codon, it will be in the P site. A new charged RNA molecule will bind to the A site. And the polypeptide bonds will form between amino acids. So for example, this one would be in P, and then another one would be in A. The amino acids up top, they will form a bond. So this complex, it will slide to the right. And the uncharged tRNA, which will be in the E site, it will exit, and then the A site will accept a new tRNA molecule. So this will continue along the whole mRNA until and then it will connect all the amino acids, forming a polypeptide. The elongation will continue until a stop codon is reached. So let's just move over here. So for stop codon, it will hit a UAA.
UAG. Or UA.
UGA or UAG. So once it hits the A site will hit a stop codon, there will be a release factor, and then it will bind to the A site. So the codon would be one of the codes for stop. And then the polypeptide will be released from the tRNA in the P site. So right here, after the release factor binds to the codon, we have a chain of polypeptide made of the amino acid. It will be released in the P site right here. So this entire process of the ribosomal unit, it will dissociate and reassemble again at the initiation process.
Thank you. Thank you. Thank you. Thank you.